Have you ever stopped to wonder if the very things that make us human, our amazing brains, our ability to think and talk in complex ways, if maybe those things come with a kind of hidden cost? Like, what if the evolutionary jumps that gave us all this also made us, as a species, more prone to conditions like autism?
It's a really provocative question, isn't it?
It really is. And today, we're diving deep into a brand new study that tackles exactly that. We're going to explore how the super fast evolution of our brains might be directly tied to why autism seems, well, so common in humans. This isn't just about understanding a condition. It feels like it's about understanding something fundamental about being human. So our mission for this deep dive is to really unpack the key findings from this paper. It's called A General Principle of Neuronal Evolution Reveals a Human Accelerated Neuron Type Potentially Underlying the High Prevalence of Autism in Humans. A bit of mouthful, but the ideas are fascinating. It's by Alexander L. Starr and Hunter B. Fraser. We want to guide you through these connections between how our brains evolved and why neurodiversity looks the way it does today. Get you informed, quickly but thoroughly.
Yeah, and it's such a shift in perspective. Thinking about autism, not just medically, but maybe from its deep evolutionary roots. And just to set the stage, think about the numbers. The WHO estimates globally it's around 1 in 100 kids identified with autism spectrum disorder.
Okay, 1 in 100 globally.
Yeah. And in the U.S., the figure is even higher, about 1 in 31 children. That's 3.2%. So these numbers really highlight why getting a handle on its origins, including the evolutionary side, is so important.
Those are definitely significant numbers. And what I find really striking is this idea that many scientists think autism, and maybe schizophrenia too, are largely, well, human things. You don't really see these sorts of behaviors in, say, chimpanzees or other primates, right?
Yeah.
Why is that thought to be the case?
Right. That's a key piece of the puzzle. The thinking is it comes down to the kinds of abilities these conditions affect. We're talking about really complex stuff like sophisticated speech, understanding language nuances, or theory of mind.
Theory of mind, that's understanding what someone else is thinking or feeling.
Exactly. Understanding intentions, beliefs. These abilities are either totally unique to us or, vastly more developed in humans than any other species. So it kind of follows that if a condition impacts those specific, highly evolved human traits, you'd likely only see it or see it in that form in humans.
That makes a lot of sense. Uniquely human skills might have uniquely human vulnerabilities. And what's amazing now is that we actually have the tools to investigate this at a really incredibly detailed level. Tell us about the tech that made this study possible.
Yeah, the technology is just game-changing. The big one here is single-cell RNA sequencing. Think of it like this. Older methods were like looking at a city from a satellite. You see neighborhoods, big structures. But single-cell RNA-seq, that's like having the blueprints for every single building. Yeah, you can see what each individual cell type is doing, what genes are switched on or off. Basically, its specific job description and how it's built, one cell at a time. It gives you this incredibly fine-grained map of the brain.
So not just broad strokes, but down to the individual neurons. And we know the brain is ridiculously complex, right? A staggering array of cell types, as the paper puts it. Plus, human brains seem to have piled up a lot of unique genetic changes pretty quickly in evolutionary terms.
That's right. Rapid evolution, especially in our lineage.
So, okay, they have this super-powered microscope, metaphorically speaking. What was the first big question they tackled about how neurons evolve? What was their starting idea?
Well, they started with something that sounds pretty logical. They wanted to figure out why some brain cell types seem really stable across evolution, while others change more. Their hypothesis was basically about abundance. They thought, okay, maybe the most common cell types, the ones that are everywhere and do fundamental jobs, are under tighter evolutionary control.
Meaning they can't change much.
Exactly. Greater selective constraint. Like if you mess with a really common critical part in a machine, things are likely to break.
Right.
Big negative effects on fitness. So the idea was these super common cells should evolve more slowly. Seems intuitive.
Yeah, that makes sense. So did the data back that up?
It did, and really strongly. They found what they called a strikingly consistent relationship. They looked across different parts of the neocortex in 6 different mammal species, and yep, the more abundant the neuronal subtype was, the more conserved its gene expression was between species. So common brain cells generally do evolve more slowly. It looks like a fundamental rule for building stable brains.
Okay, so that's rule number 1. Common cell types usually change slowly. That's cool in itself. But the really kicker comes when you use that rule to find the exceptions, right? What happens when you apply this to the human brain and look for cells that don't follow the pattern?
Precisely. That's the power of finding a general principle. It lets the outliers, the exceptions, really stand out. And that's what they did. They looked for neurons in humans that buck this trend, and they zeroed in on one specific type, layer 23 intratelencephalic excitatory neurons, or L23IT neurons for short.
The L23IT neurons. Okay, tell us about those. What do they do, and why are they important for us?
Right, so these L23IT neurons are super important. They're actually the most abundant type of neuron in the outer layers of our cortex. Think of them as major communication hubs. They're essential for connecting different areas of the neocortex, sending information back and forth. And this communication between areas is thought to be absolutely key for higher-level human abilities, planning, complex reasoning, language.
The stuff that makes us, well, us.
Pretty much. And given they're so abundant and so critical, according to that rule we just talked about, you'd expect them to be highly conserved, slowly evolving in humans.
Just like in other mammals. But I'm sensing a but coming.
Exactly. Here's the twist. In the human lineage, these L23IT neurons evolved exceptionally quickly compared to our ape relatives. They're a massive outlier. These incredibly common, crucial cells should have been stable, but in us, they went through rapid changes.
Wow. Okay, so that really breaks the mold. When you see fast evolution like that, is it like evolution actively pushing for those changes because they're good? Or is it more like the constraints just loosened up and changes happen by chance?
That's the key question. Positive selection or relaxed constraint? Was it a deliberate upgrade or just, stuff happening? The study found pretty strong evidence pointing towards positive selection.
Ah, so an active upgrade. How did they figure that out?
They looked at how much the gene expression in these cells varied from person to person, and they found that L23IT neurons have the lowest amount of variation between individual humans compared to other cell types. Yeah. If it was just relaxed constraint, you'd expect more random variation accumulating. Low variability suggests these changes were actively selected for, fine-tuned, and kept consistent because they were beneficial.
Okay, that makes sense. Like dialing in a specific setting. So these crucial communication neurons in our brains underwent this rapid, selected for evolution. What did that actually mean at the genetic level? What changed?
And this is where the connection to autism really snaps into focus. This rapid evolution wasn't random. It involved very specific changes in genes already known to be associated with autism spectrum disorder. Specifically, they found a strong and specific enrichment for lower expression of high-confidence ASD-linked genes in these human L23IT neurons.
Lower expression. So these genes were turned down.
Exactly, dialed down. And importantly, this wasn't just one or two genes. It was driven by what they call polygenic positive selection, specific to the human lineage.
Okay, polygenic positive selection. Let's break that down. Polygenic means many genes.
Yep, lots of genes, each maybe having a small effect, but working together collectively, like an orchestra, not a soloist. And positive selection, again, means these combined changes were actively favored by evolution because they provided some kind of advantage. So evolution effectively coordinated the turning down of a whole suite of autism-linked genes, specifically in these L23IT neurons in humans.
That's incredible. A coordinated dialing down. Did they see anything similar for genes linked to schizophrenia, given the overlap sometimes mentioned?
They did, yeah. They found similar evidence pointing towards downregulation of schizophrenia-linked genes too, although the statistical signal wasn't quite as strong as for ASD genes. But it fits with the known overlap, genetically and symptom-wise, between autism and schizophrenia. It hints that maybe similar evolutionary pressures were involved for both.
Okay, so the genetic data points to this selected for downregulation in humans. But how do we know for sure this is genetic, baked into our DNA, and not something influenced by, I don't know, the environment or development after birth?
Excellent question. How do you disentangle nature and nurture here? They used a really clever technique. Human-chimpanzee hybrid cortical organoids.
Hybrid organoids, like mini brains in a dish.
Sort of, yeah. They grow these small clusters of brain cells from stem cells in the lab. But the key is, these were hybrid cells containing both human and chimpanzee DNA within the same cell nucleus. So you have both the human version and the chimp version of genes sitting side by side in the exact same cellular environment.
So that removes the environmental difference.
Exactly. Any difference in how much the human gene versus the chimp gene is expressed has to be down to the DNA sequence itself. And what they found was a clear bias. The human versions, alleles, of those ASD-linked genes were expressed at lower levels than the chimp versions, even in the same cell. This strongly confirmed these are cis-regulatory changes, differences written into the DNA code itself that happen along the human line. It's genetic.
That's Pretty conclusive evidence then. It's built into our human genome. But okay, this leads to the big why, doesn't it? Why would lowering the expression of genes linked to autism actually be a good thing evolutionarily? Why would that give our ancestors an edge? It feels a bit backwards.
It does seem paradoxical at first glance, for sure. And the paper acknowledges the exact why is hard to pin down definitively, but they offer a couple of really plausible hypotheses. One idea is that this downregulation might have helped enable our much slower, extended period of brain development after birth compared to chimps.
Our long childhoods, essentially.
Right. That longer runway for development allows for way more learning, brain plasticity, building more complex connections, basically setting the stage for more complex thought. The second main idea revolves around language.
Ah, the superhuman skill.
Exactly. Speech production, comprehension. These are hugely expanded in humans and are often areas impacted in autism and schizophrenia.
Oh.
So perhaps turning down these specific genes was part of the genetic toolkit that allowed our unique language abilities to flourish. It might have been part of the cost of developing language.
So potential benefit, like a longer development window, maybe better language. But if these changes were beneficial, how does that lead back to a higher prevalence of autism in humans? It sounds like a trade-off.
It absolutely sounds like a trade-off. And the researchers proposed this really interesting model. They call it the ASD expression threshold model. Think of it like a volume knob for these autism-linked genes in the brain. In our ancestors, maybe chimpanzees, that knob was set at a certain level, let's say level 10. Through human evolution, that polygenic positive selection effectively turned the species' typical volume knob down to, say, level 7. Now, this new baseline level 7 is what gave us those advantages, slower development, better language, whatever the benefits were.
Right, so the average human setting is lower now.
Yes, but it also means that as a species, we're now operating closer to a hypothetical threshold, let's say level 5, below which autism-related traits start to emerge. A great example they use is the gene DLG4. It codes for a really important protein at synapses, the connections between neurons.
Okay.
Human L23IT neurons express DLG4 at a level about 2.5 times lower than chimpanzee neurons do. Now, that lower baseline in humans doesn't cause autism by itself, but it means a relatively small extra nudge downwards. Maybe losing one copy of the gene or some other genetic factor, or even an environmental influence could push an individual below that level 5 threshold.
Whereas it's the same nudge in a chimp, starting from level 10, might only bring them down to level 8, still well above the threshold.
Exactly. So the evolutionary shift that benefited the species as a whole also, inadvertently perhaps, made individuals within the species more susceptible to falling below that crucial threshold if other factors come into play.
That is, wow, that's a really profound idea. So the very genetic shifts that sculpted our unique human cognition, our language, might have simultaneously brought the whole species closer to this tipping point, increasing the chance of neurodevelopmental variations like autism.
That's the core implication, yeah. It suggests that the architecture that makes us uniquely human might inherently include the blueprint for our unique forms of neurodiversity. Like Alexander Starr, the lead author, put it, and I'm paraphrasing slightly, some of the same genetic tweaks making the human brain unique also made humans more neurodiverse.
It really reframes things, doesn't it?
It absolutely does. It links our greatest strengths and perhaps some of our unique vulnerabilities at a really deep evolutionary level.
Okay, so let's just recap this deep dive. We've explored how this one specific type of brain cell, the L23IT neuron, super abundant, vital for communication across the cortex, underwent incredibly rapid evolution in humans. This evolution seems to have been actively selected for and involved turning down the expression levels of a whole set of genes linked to autism.
Yeah, driven by polygenic positive selection.
Right, and this wasn't just random, it was confirmed using those fascinating hybrid organoids. It seems this downregulation might have given our ancestors advantages, maybe in brain development speed or language capacity.
But the flip side, the trade-off, is this expression threshold idea. By lowering the baseline expression of these genes as a species, we might have moved closer to a point where smaller additional genetic or environmental hits could lead to autism spectrum conditions.
So it really paints this picture of our evolution being this complex balancing act. What makes us unique might be inseparable from what makes us vulnerable in certain ways.
And I think this offers such a powerful lens for thinking about neurodiversity. It moves away from seeing autism as purely a disorder or a deviation. Instead, it suggests it might be a fundamental, maybe even expected, outcome of the evolutionary path that made us human in the first place. It's woven into our species story.
That perspective is incredibly important for how we understand and support everyone in our society. So as we leave you to think about all this, here's a final thought. If the journey to our unique minds and voices involves such a major, rapid overhaul of our brain's most common neurons, potentially increasing the odds for conditions like autism, what other unforeseen evolutionary trade-offs might be lurking in our biology? What other aspects of human variation, maybe even things we consider illnesses or weaknesses, might actually be tangled up with the very traits that define our success as a species? What does this really mean for how we understand ourselves and maybe even how we think about our future?